All right, now we can get started. Welcome everyone to the Playbook Workflow Builder Workshop. I'm Hisu Kim and I'm Systems Analyst in the Maya Lab and a member of CFD Data Resource Center. The plan for the workshop is for Dr. Avi Mayan to provide a brief introduction, and then Daniel Clark, the lead developer of the Playbook Workflow Builder platform, will demonstrate how to use the system first, and then in the second part of the workshop, he will go over how to develop workflow components that could be added to the platform. So I will invite Dr. Avi Mayan to give the introduction.
All right, thank you, Isu, and thank you everyone for coming to the workshop today. I'm just going to give a brief introduction, and then Daniel Clark will conduct the workshop. I'm just going to place the project in the context of some of the other projects that we had over the years. So if you can pass into the next slide. So in 2016, we published this paper in F1000 Research, and this is Jupyter Notebook that performs RNA-seq analysis. And this is the time that Jupyter Notebooks sort of like arrived, and we realized that they can really be used as a publication, a parameterized publication. So here, this RNA-seq analysis pipeline was applied to some Zika virus infection of cells from a published study. But the idea was that people can take that notebook and change it with their data and run it and get a paper that just is applied to their data. Next slide. So advancing on that concept, 2 years later, we published this paper called BioJupys. And with BioJupys, you can come in with your RNA-seq data. You can upload it, and then you select the samples that you want to compare, as well as the tool that you want to use in your report. And then once you press submit, you get a report that analyzes your data that also resemble publication. So this was an advancement because here it was more like a TurboTax for data analysis. And this is still widely used app, but is only for RNA-seq. And so it's very specific for this project. Next slide. So then when Daniel joined the lab, he developed sort of like the successors of BioJupys that made it more general. So now we can apply the similar approach to build very fast, lightweight bioinformatics application by converting Jupyter Notebooks into a full stack web-based application by putting in the notebook a few global variables that turn into a HTML form once you compile the appiters with an SDK. And so far, if you can move on to the next slide, we published over 100 of those appiters. And those are also widely used by the community for various types of bioinformatics analyses. Next slide. So today, the subject of the workshop is this Playbook Workflow Builder app, this new app that is the successor of Appiters, also headed by Daniel Clark. This is CFDE funded project. It's a partnership that includes other members of the CFDE. And we all been working on this together for the last 2-3 years. So you will hear a lot more about this platform, this exciting new platform. If you can go to the next slide, please. This is really an evolution over the past 10 years. We were members of the BD2K program, and then that program turned into this DCPPC program that is now the CFDE. And the apps, the Jupyter Notebook that was published in 2016 is now a lot more elaborate application that is really transformative in this field of bioinformatics and infrastructure for bioinformatics. So I just like to thank the NIH for funding, for the other members of the Playbook partnership, and the support from the NIH for continuing these projects. So now I'll pass it to Daniel, who will give the workshop. It's divided into two parts. So the first part would be about demonstrating the platform from a user perspective, and then the second part will be more from if you want to contribute to extend the platform, we have a way for you to create your own workflows as well as contribute to this network of microservices that are forming this platform. So Daniel, go for it.
Thank you, Dr. Mayan, for that introduction. And as he mentioned, I'll be talking about the Playbook Workflow Builder platform. I'm a data science analyst in Dr. Avi Mayan's lab, and also a member of the Common Fund Data Ecosystems Data Resource Center. And we're coming out of the Icahn School of Medicine at Mount Sinai. Just to recap from what Avi mentioned, we are essentially evolving this platform to go beyond just data notebooks, and we're trying to now enable people to build their own appeters, if you will, these playbooks, without having to code at all. So before, in these other platforms, you're reusing a pipeline that someone else created with your own data. Now we want you to be able to build your own pipelines. So to do this, we first started by cataloging the Common Fund Data Ecosystem landscape of APIs that are already available and programmatically accessible. And we took these various APIs and we codified them into this component, which you'll see later. And each component has some kind of inputs and some kind of outputs, and that is what really allows you to go from one to the other in a seamless way. And in order to drive the selection and insertion of these different components, we developed a wide range of different use cases that would span data from many of the different sources of programs that are in the ecosystem. And we came up with these types of biological questions we could answer by hopping through them. And each of those use cases we implemented as playbooks on the platform, stepping through these different components. So the platform looks like this. This is the landing page, and I'll just give a brief overview of how to use it. And then I'll actually show you in real time how you can use this platform. If you would click one of these inputs, we have, you can start with many different things, but we'll start with a gene. You're then granted with a form, and you can put in a gene in this case, but in the other cases, if it's some kind of file upload, you can upload a file here and whatnot. Once you've put in this gene and clicked the submit, what you're shown in this next step are the endpoints that are in the platform that are compatible with the gene as input. So we can see that we can use the Links L1000, we can do a MET gene search for metabolites, or we can resolve regulatory elements from the Common Fund linked data hub. And we can also query GTEx for median tissue expression. So if we would click this, we, in real time, actually query the GTEx database, and we get this output, which has various tissues and a z-score showing significant expression of that particular gene that we started with in that tissue. And from here, we can do several other things. We can just select some of these tissues to process in some other way, potentially sending to some different API, or more simply, we can build some kind of visualization. So here we click on the vertical bar plot, and when we do that, we get a nice plot that's showing the table that we had before. If we click on this top right icon, which is the report mode icon, we're able to get this vertical look of the workflow. And this is a shareable entity that can be stepped through completely. So this is really like what the output you would have got from an appeter. You get this type of summary, which is based on the workflow you created. And we can use GPT to actually augment this summary. We get the input that we started with, and actually people who receive this workflow can actually put in their own input, and the workflow will re-execute all of the steps beyond the input with the new input. We can also name this workflow, give it a title. We can manually put a description, and then we can save this to our account. And we can also export the workflow with a variety of means. And one of those is the biocompute object, which is a way of documenting this particular workflow and for citation purposes. And we can also export this common workflow language bundle, which makes this workflow compatible with other workflow execution systems. The playbooks that are published by either you or by us appear in the publish section of the platform, and these contain basically implemented versions of the original book of use case libraries that we assembled, and a slew of playbooks that are being built by the community and hopefully by you today. So now I'm going to show a hands-on demo using the platform, and I hope this will better introduce you to the platform. So if we open up the Playbook workflow site, we can then come here and we have the options again, but instead of doing the one that I've been showing with the starting with a gene, instead we're going to upload a gene count matrix. So when we do this, we actually have to sign in to upload files into the platform. So I will sign in. I recommend that you sign in with your ORCID. So I will do that. Let me get my ORCID. Sorry. Once I've signed in with ORCID, we can then see the input form. And this input form lets us choose files from our disk. But I'm just going to use the example for now. So if we click example, this will upload this data file that has melanocytes and melanoma samples across all genes. This is a gene count matrix. You can download these to see what it looks like on your disk. But once you've uploaded a gene count matrix, you can then see what you can do with this. So we have a wide variety of different things we can do. We can normalize the count matrix. We can perform various visualizations. One useful thing is performing differential expression analysis. So to do that, we actually have to label the samples in our matrix. So if I click on manual labeling of samples, we can select basically what are the controls and the perturbations of our experiment corresponding to each of those columns in our data file. So if I make melanocytes the control and melanoma the perturbation and click submit, once this loads, we basically have these samples now annotated. And if we extend from here, we can do certain operations on this annotated version of our gene count matrix. We can perform differential expression analysis now. So I'm going to perform Lima-Voom, which is a pretty popular differential expression analysis. And while that's running, I also want to show that you can perform a PCA graph. So you can basically do PCA on this data file. And so if I click on that, we'll see that once these load, we'll get it. So here is the PCA graph. We can see that our control samples in blue appear separated in PCA space to the perturbation samples in red. And we can see that this part with the differential expression analysis has now turned green, which means that it's ready. So we can click on any of these nodes to move around to see what we have. Since this differential expression analysis is complete, we can see that every gene has a p-value, an adjusted p-value, and a log fold change. With this data, we could download this table to process it, but we can also continue checking things from the platform. So one thing we can do is actually get these, just extract the genes and the scores from the matrix because we have other things that are compatible with this. When we do that, we can see that some things we can do are do like a signature search in Geo or in links. And this will find signatures in these databases that like match our experiment. Another thing we can do is just take, say, the top 10 or top 50, top 100 genes of this table. And when we do that, we're able to get a set. So essentially, we're just taking the most upregulated genes from this differential expression analysis. And with this, we're now compatible with a whole slew of different gene set enrichment techniques. We can apply, for instance, transcription factor enrichment analysis to enrich our gene set for different transcription factors. We can perform kinase enrichment analysis. And we can also go to metabolomics, go to signature commons links to get drug perturbations. So I can actually do that with the gene set. And when we do this, once this loads, we'll see that what we've now found are drugs which are mimicking or reversing the expression. Oops. The demo gods didn't like that. But I think that we extract. Okay. So it seems the site had some error, but we're still in fact able to extract the drugs. So we weren't able to visualize it, but these are the drugs that actually are mimicking or reversing the expression of those top genes. So with that, I'll end it here, but we can go back here, we can do other things. At every step of here, we can extend and fork this further. And once we click on this view report, what we see is the whole pipeline in this vertical section. So we can see step by step, we uploaded the matrix. By the way, we also have this complete summary, which cites the different methods used. We used Lima and Voom. We used these transcription factor and kinase enrichment analysis. We used the signature commons links data and metadata search engine. And again, every step is shown, and we can then like share this with someone else. So how do we share it with someone else? Well, because I'm now logged in, I can save this, so I can give it a title. My first analysis with Playbook. And then if we save this, we can now click on share link. And if I copy this link and I would give it to someone else or visit the page, you'll end up right back here. Another user that would come here could in fact click on expand from this step to continue the workflow, for instance. Another place that you can find your published workflows is if you go to your account settings and then you go to Playbooks, you'll see that the titled Playbook appears here with your workflow, and you can go back to it if you needed to get it again. So that I think concludes this particular demo.